Yes.
So Augustine is works at BIIF. I have mentioned this facility several times during the course. BIIF stands for Bioimage Informatics Facility. It's a facility at SciLife, which has 1 foot in Solna in Stockholm and 1 foot at Uppsala. And he's an image analyst there, and they are really great. They develop software and algorithms, so they work in research about image analysis, but they also, on the facility side, they also support people. And so that means that they know all the latest tools, and they will find the best possible tool that to help you build your pipeline. So maybe Augustine will present the facility afterwards. I warmly recommend contacting them. Please, Augustine.
Yes, thank you for the introduction. Yes, as Sylvie mentioned, we're part of NBIS, which is the National Bioinformatics Infrastructure in Sweden, and within SciLab. And I'm going to give you a tour de force on image analysis. So I'm going to mention many things. Some things you already heard from Sylvie before. And there are more slides that are shared with you than the ones I will show right now, so that you can go through them afterwards, because you're a bit repeated with other things. So yes, we are the Bioimage Informatics unit. We give support, training, and tools for image data analysis. We use state-of-the-art analysis with this image data, such as computer vision, machine learning, bioinformatics, and we also help in deploying computational methods for your research. So this is us. Most of us are based in Uppsala. I also have an office here in Solna, so I spend some time of the week in Uppsala, some time of the week here in Solna, and yeah. So let's get right to it. So what is bioimage analysis? So the definition is this one. Bioimage analysis is the process by which one extracts quantitative data or information from scientific images. And I want to highlight two things here, because we need, one is quantitative, because we need to take numbers from this and something that tells us something about the sample. So, and the other word I would like to highlight is scientific images, because we need to be able to reproduce the acquisition of the images, and they have to be clearly documented and understood for other scientists to be able to reproduce them as well. So here we have an image. These are nuclei acquired with a fluorescence microscope. And I would like to, if you can open the chat and write how many nuclei do you see in this image? Can you just quickly count them and write it on the chat? Other questions we might wonder is, okay, can we estimate the intensity per nuclei, or how big they are? How can we do these things? Because we could do it manually, for example. We could use some tool to draw the regions of interest around the nuclei, or we could threshold on intensity and take the values from the output table. So let's check the answers on the chat, because one thing I did not mention here is what do we do with the nuclei that are on the edges? Because I get some people might have counted them, but others didn't, because I mean, this one you just see a very small part. Should you count it? Should you not? So this would have been part of the bioimage analysis protocol. This should have been described and defined beforehand, before we even started analyzing the image. There are other questions that we could answer, like, I mean, do we always need to do bioimage analysis? Can't we simply show some image and say something about it? Like, for example, here we could wonder what happens with the localization of a protein. We have two cells here, and we reason that the cell B has been treated with a drug. And we believe that this drug has displaced the protein from the nuclei to the cytoplasm, because we see that the cytoplasm is brighter and the nuclei looks dimmer. but are we sure which nuclei is brighter here? So here we have one of these typical optical illusions that we can find. It's not enough to just show an image. If we were to quantify this one, we would have noticed that the nuclei is brighter. It's the same intensity in both. So maybe there was more protein in cell B that happened to be in the cytoplasm. Similar questions can happen with respect to sizes. We say, okay, we add some drug to these cells and a particular cell type starves, leaving the one in the middle starves and leaves more nutrients for the one that are surrounding, so they grow larger. And the cell in the middle gets smaller because it's not acquiring all these nutrients. But after analyzing the sizes, we notice, okay, this is again another optical illusion. The cells are the same size. It's just the other cell type that was affected by this drug. So what are the characteristics we want of a bioimage analysis protocol? So the previous example already led us to one key characteristic. It has to be it's supposed to be quantitative. If we want to specify the intensity of each cell or the size of them, we would have noticed in the previous examples that we were saying something wrong just by looking at the image. Next, we want our observations to be objective. We could ask 3 different observers to segment this tumor outlining in these images, and each of them have a different appreciation of what the border is. Some are a bit more broader, others focus only on the dark parts, and they always get a different volume for this segmentation. So if we apply the same bioimage analysis pipeline or protocol, the results should be objective. It doesn't matter who is running it. We always get the same number. Next, we would like, and following this, we would like it to be repeatable. If we start from the same raw data, we want to be able to end up with the same output. And we would also like it to be reproducible. So one way of doing this is what we have done before. Just describing in a step-by-step basis in materials and methods. We can also write a script like this one to share it, and then publish it with our publication. But something important that we have to highlight here is that even if the pipeline does not seem to be yielding the same result, as you can see here, we try to count the number of tangerines in this image, and we see the pipeline says there are only 2 tangerines. We repeat the analysis and we always get the same value. This means it's reproducible. And the good thing is that science has to be reproducible, because we might not agree with the criteria I used during this analysis, but then you could totally run the same pipeline on your images, or do the same analysis and say, okay, I do not agree with your analysis because I get this result, but you are able to reproduce my results, build on top of them, and then we can start discussing. If you're not able to reproduce my results, then you might even start doubting what I was doing before. And finally, the last characteristic bioimage analysis ideally has is that it should be reliable. So the previous example I showed that the measurement was not perfect. We were getting the wrong number. But while developing the image analysis pipeline, we should aim for reliability, and we should characterize and understand its limitations. So ideally, we should have test cases, we should be able to validate, and we should be able to quantify the accuracy and how good our pipeline is, but also what are its limitations. There's always going to be limitations when we develop a pipeline. And we will see in the future how to work around this tangerine segmentation problem. So there are more advantages. I mean, if these previous regions were not enough to convince you that bioimage analysis is key, there are a couple more. For example, nowadays there's a lot of high throughput image acquisition, and they generate tens and hundreds of gigabytes of images, even terabytes. I will show you a couple of projects that we worked within our unit. As example, here we have a 3D imaging with light sheet microscopes. This easily generates gigabytes of images hundreds of gigabytes per sample. And then we start wondering, well, we should add more samples to add some statistical power. We should see more cases. So it increases very fast. We could also have time series experiments, and then we have several images. We can acquire several images per second or several images per minute. Do this for many minutes, even hours or days. And this will also yield thousands of images. Drug screening experiments, where several wells are containing cells and they're treated with different drugs in each well or combinations of them. There's also in pathology, samples can take up to several millimeters in height and width. So we might be unsure which region is of interest. We would like maybe to acquire the whole slide, and then we end up tiling the image and stitching them together. So again, this is a lot of data. Some other cases we might have, in some cases we have 2 or 3 antibodies or reporters that we just place in 3 channels and it's okay. But in many other cases, we need several antibodies to define a specific cell type. And the newer technologies allow us to do several cycles of staining and washing on the same tissue. So we can easily end up with 20 channels. And we could also combine different modalities. We could combine the modalities I mentioned before. We can go into spatial transcriptomics, for example, and then we would like to detect the position of RNA transcripts within our sample. We generate tons of data very easily. And the advantage of bioimage analysis is that even if the data sets increase in size, at some point we will have to do it automatically, because on one hand, these analyses are faster than humans, but they're also less biased than humans. Where do we start when we want to design a bioimage analysis pipeline? How do we know we even need bioimage analysis? Well, what will we be using? Well, as it was mentioned before, and this is a very important thing, we always start from the scientific question. What is the scientific question? And it has to be clearly defined. It's not, you cannot ask something like, okay, is drug A better than drug B? What does that mean? What is better? We could ask something like, does drug A inhibit growth of cell type M, while drug B lets them keep proliferating? This is better, because now we know what we want to quantify, and then what we want to image. So if we know what we want to quantify, we would like to see if cell type M is proliferating more or less, then we can start reverse engineering from the quantitative information that we want to inform this hypothesis, which image acquisition we should aim for. What do we need to have inside our image? So this way, now we know what we have to image and how we would like to image this. We already know, okay, we have to image this cell type M. We need samples that are going to be treated with drug A, some with drug B. We need a couple controls as well. And this means we already can design the experimental procedure. We can deal with image acquisition. But after performing some of the experiments and doing a bit of the image analysis, we will have some quantitative results that we can feed into our statistical model or mathematical model, whatever we're using. This could even be a toy model of but it has to reflect our understanding of the system and what is going on here. And this will help us understand, okay, did we think through our question? Did we think through our image analysis? Do we need to quantify something else? Do we need to improve image acquisition? So all these gears feed back into each other and help design better experiments, which are more informative and answer better our hypothesis. Yes, so, and just as an example, the clearly defined question, it could be a biological question, but there are many bioscience projects that start with questions in any of these gears. So some people even say, okay, I have a particular mathematical model to which system I can apply it to. That means I should be acquiring images of this type, and then the image analysis should yield this type of result. And this keeps feedback into each other gear and keeps generating different projects and more questions. So how is a typical analysis workflow? How do we see it? Well, we always use, you can start designing it from the raw images. As we discussed previously, we need to understand first how these digital images work, what is the file format we're going to use, image data and metadata. There's been a couple lectures about the digital image, so I might go fast on some of these slides, but I want to refresh your memory. The next step is called pre-processing. It can include several steps of correction, such as background correction, illumination correction, maybe noise suppression of the images. Then comes the tricky part, and segmentation is about dividing the image into different regions, the ones that correspond to the objects of interest, and the one that corresponds to background pixels. This is usually very tricky and often appears in many image analysis workflows. We can have a refinement step here to clean out salt and pepper noise, for example, and then what is called instant segmentation to distinguish between the different objects that are in the images. And when we have all these objects identified, then we can proceed with quantifying and extracting features from these images. Not every project has all steps, but this is an overview that it's often found in many of these projects. And I will also use this slide as an outline for the presentation so that we go through the different sections separately. Let's start by the raw or digital image section. If you need to stop at any time or have further questions, don't hesitate in telling me. I might go a bit fast because this is you already know about these things. So what is a digital image? So you already know that it's a matrix of numbers, and each element in this matrix is called a pixel, and it represents the intensity measured at this specific position. It could also be a voxel if we were talking about 3D images. We can take this monochromatic image, for example, and we observe some structures that correspond to the cytoskeleton of the cell. If we zoom in a lot, we start seeing this sort of squares. These are the actual pixels. And remember that these edges are artifacts of how we are showing the image. These edges are not there in real life. And you can look at this digital image in different ways. So you can show all these intensity values in a matrix. You can see that the position refers to a position in the real world. And how are these pixels defined? So where do they come from? Well, you already know that they come from the image generation process of how you acquire the images. Some other key characteristics is, for example, the resolution versus the pixel size. And this is something that we've discussed a lot during the course, and it's yeah, I know it's hard to understand, but by this point, I think most or all of you already understand this. These pixels are referring to positions, and we can see here, for example, that in this particular image, the pixel size is 3.3 micrometers, so they're quite big. I mean, we cannot see much. So if we were to acquire the image with a smaller pixel size, then in this case 0.8 micrometers, we start seeing some structures and some things there. And we can even acquire the image with a 50 nanometer pixel size, and then we see more detail inside the image. But did we have to go that far? So this pixel size is a property of the digital image. But resolution is a property of the system that we were using to acquire the image. In this case, for example, we can see two images where we acquired them with different pixel sizes. And sorry, we acquired the same pixel size, but with different resolution. So we see even though the pixel size is the same, if we change the imaging system, the resolution will be different. And in some cases, we will be barely able to distinguish between these two structures over here. So at this point, you are probably thinking about the Nyquist criteria. So we know that positions are discretized in our image, but what about intensity? Because these values are also discretized in an image. We have at some point our analog to digital converter. So we discretize the intensity values or the number of photons we get from the image into some intensity value. This is called bit depth, and it's also represented by the number of gray levels you get in an image. We could have chosen, for example, a bit depth of 1. This means we have 2 to the power of 1 possible intensity values. So it's either a 0 or a 1. And if we use grayscale to show this, we will see in this image, for example, either there's some signal there or there's nothing. So we get either a 1 or a 0. We could go a bit further and say, okay, let's take a bit depth of 2. Then we have 2 to the power of 2. That means we have 4 possible gray values here. And if we check the image, we start seeing that some of the structures that were within this head that we were imaging, they have a different intensity or a different gray color. But we could have gone all the way to a bit depth of 16, for example. Then we get over 65,000 different values, and we can see a lot of different structures with different textures. There's a lot more information here. So all in all, we can increase the bit depth, and this will allow us to distinguish between different intensities. But remember, this is not completely free, because the bit depth relates to how much space in disk each pixel will occupy. And remember that if you have an image that's 2000 by 2000, then we're talking about millions, and then the more you increase the bit depth, the more space it will occupy in disk, and you have to learn how to deal with more data. So we discussed mostly about monochromatic images, but what if we have color images? So the monochromatic images, we have just a single matrix of values. In a color images, we need at least 3 to represent the red, green, and blue colors. And so this way we can actually decompose our image into the red channel, the green channel, and the blue channel. In this case, it might not help us too much because the information is kind of mixed. We kind of see the nuclei in all 3 of them, and then, yeah, it's hard to interpret this. But we can apply something called color deconvolution or spectral mixing of these dyes, and then we get the hematoxylin on one channel and the eosin in another one, and we can see mostly the nuclei in one of them, and then the eosin in the other one. And the last one is kind of what was left. It could be attributed to noise in some cases. So in fluorescence microscopy, we usually have one channel corresponding to different ranges of wavelengths that we were acquiring. And these are all saved in different matrices, similar to how I was showing it before for a color image. So this means that each channel has a gray level representation, or can have a gray level representation, and that they are saved in independent matrices. So there's no need to be deconvolving between the different channels. So here we have one channel for each of the different structures, and we could also use cyclic stainings to have more and more channels, and this means we have more and more matrices for the same image. In this case, we're only working with these 3, but maybe red, green, and blue were not the best colors to choose. So we can grab one of the channels and say, okay, we're going to use cyan instead of green, or maybe we want to show the background and also the really bright intensity. So we go for this LUT is called Inferno, and these LUTs are called our lookup tables. So what they do is we change the range of colors to highlight different things. And my personal favorite, and that you have seen quite a lot of times in the course, is the high-low, which actually is a grayscale channel for most of the values, but it will show in blue color for whatever is 0 or below 0, and in red for whatever is at saturation. So remember that these lookup tables define how the image is displayed, and you shouldn't be changing the original values. So this is mostly to change how you see them, how you see the images. And if you can also merge all these channels together, but then think about the LUTs you're going to choose for each of the channels, and consider people that might be colorblind as well. So there's another thing that will give us information about intensities, and this is histograms. We went this a couple times during the course. and we can get basic measurements from them, such as the mean, standard deviation, the minimum and maximum. Pixel order does not affect the histogram, as you know, and you can already see, for example, how different a dark field image, or sorry, a image from a fluorescence microscope is from a bright field image, because the pixel intensities of the background often occupy most of the histogram. So you can see in one case it's all skewed to the left, and the other one is all skewed to the right. You can play around with brightness and contrast to make the image look better. This we have seen. Remember that you can choose reset to go between the minimum value and the maximum value of the image. You can choose auto for that. Just remember that try not to click on apply, because whenever you click on apply, you are changing the values from the image. So in this case, I had this image, I played around with the brightness and contrast, I clicked on apply, and then I ended up with a completely different histogram. So I briefly mentioned something about our image dimensions. Usually we see them as two-dimensional. So here we have one image that has 408 pixels by 406, but we can easily go into 3 dimensions if we consider the different channels. And this third dimension could have as many channels as needed. So in this case, it's 3, but it could have gone to 20, 50, or whatever. We can also think about, okay, maybe we do a seed stack of our sample, so then we have an extra dimension. In this case, it's a seed stack of 15 steps and 4 different channels. Or we could even add that time dimension. So we take images every couple of minutes or every couple of seconds, and then we end up with 5 dimensions. This is the highest number of dimensions we could be having in an image, and that's it. So great. We have discussed a lot about images, and they have a lot of information, but it's also quite hard to just guess what's in an image just by trying to open it, because we will end up with a matrix of pixels. That's why we have the image metadata, and this metadata can be of experimental type, so it could contain probes or the treatment we used, the sample that we were imaging. It has information about the acquisition, what microscope we use, what it was the pixel size, filters that we were using. It could also have analytic information, such as we ran some deconvolution, we ran some stitching. But be careful, because not everything is automatic. Not all the metadata is saved by default. So it requires someone to configure your microscope. They're usually configured, so remember that there are people working on this. But if you get a microscope in your lab that nobody has worked on, it might not have all these things configured. Yeah, so most of the metadata is stored and is saved by if you're using some proprietary format from the company that sold you the microscope. So we have ND2 for Nikon, or LIF for Leica, OIF for Olympus, ZZI for Zeiss, etc. But there are other general file formats that you've also seen yesterday. JPEG, for example, usually has a lossy compression, so you will lose information if you save it on JPEG. TIFF is often a lossless format. It could be lossy, but it stores metadata. So you should be able to store in the metadata what you have done with this image and what each matrix is. And PNGs are usually used for transferring or displaying things, but you cannot really do stocks with them. So they're nice for showing, but not for many other things. There are file formats that are usually fine for figures, like bitmaps, for example, or vector images like SVGs or PDFs. As you've seen yesterday, you can have them of a raster type, so then you see the pixels if you zoom in a lot, or the vectorized ones. You don't really see pixels because you're saving the lines, the geometric lines. And we have open microscopy environments. So you've seen yesterday people from OME, and you can have OME TIFFs, for example. So they have a strict way of how metadata should be saved. And there's also what's called next generation file format, which is often called ZAR, or OME ZAR TIFFs. In this case, you're also saving them and on a different way, so that if you have a huge image, it's easy to find it and find the pieces you need in disk in order to show it in your computer. Because most of the times you will have to load the whole image to show it on VG, for example. But with if you really know where the piece of information is to show the image, you don't have to load everything. And this is great if you're working with really big images that you cannot fit on the memory of your computer. So how do we open them? Because now we ended up with a ton of file formats that's, yeah, it's gonna be very complicated. Then you might wonder, okay, do I need a piece of software for each of the different file formats? Well, we do have the bio formats, and it's also a plugin that comes with Fiji. So most of the formats here you can open them with Fiji. You can open them with this bio formats. So remember to always keep the original files and the raw data. Yeah. So let's jump into pre-processing. So let's say we have already acquired our raw images. We ended up with a ton of images. And before attempting to segment them, we would like to clean them up a bit. So it's not always necessary, but it's a good practice to do it, especially if you have to do intensity quantification. And here we'll mention a couple of different image artifacts. Some of the image artifacts I'll mention can be corrected during image analysis, like illumination inhomogeneities or background. Some are very difficult or even impossible in some specific cases. Bleed through sometimes, registration sometimes, well, dirty samples. And some others are actually impossible because we lost information during the acquisition, such as saturation or wrong pixel size. So let's say we want to estimate our background from our image. We could maybe, if we don't have any other information, select a region that should correspond to background and estimate what is the level of background in this image. We could also characterize our background from our microscope by acquiring images without the sample. Or we could apply some algorithms like rolling ball that we will see afterwards. But to do this, we need to be able to understand what image operations are. And these are designed to modify or enhance some image properties, and they often take one or more images, they apply a specific function, and then you get an image as an output. We can separate them in per pixel operations. So they take a pixel, do some calculation, and give you a value for that same pixel, or neighborhood operations, which take a group of pixels surrounding a central pixel and give you the value that should be replaced there. Or we could also categorize them depending if we are working on the spatial domain, in this case, the image per se of a neuron, or we could convert the image into frequency domain, as you've seen with a Fourier transform, and work on the frequency domain. So let's say we decide to take, we want to do some quick analysis, or it's just to make a fast estimation. We select this specific ROI, we found the background value here, and we say, okay, it's this specific value, and we just subtract that value from every pixel, and we get this image in the end. We see that the background now is all completely blue. This might be as a quick fix. Or we acquired several different images from background. And the first thing we will do is per pixel estimate the median or the mean intensity, so that we end up with a single background image. And that background image is going to be subtracted from the image we acquired, and we end up with the image without background. So this is another way of doing it. This is it requires you to characterize better your microscope. And let's say we have another problem now. We acquire their images and the illumination was not homogeneous. So it often happens that there's a region of the image that's quite more bright than another part. We can characterize this by using some homogeneously fluorescent slide or sample. We can acquire several different images and, as before, calculate the mean per pixel. And then we use this after we have to subtract, remember to subtract the background from this one, but then we can divide our image by this. Or there are some other cases where we acquired so many images, or like tiling, for example, or time lapses. So we could run specific algorithms that estimate the illumination profile from the images we acquired. In this case, for example, it's an algorithm called CIDRE, or there's another algorithm called BASIC that we will see during the workshop. And you see how the illumination has been corrected after applying this algorithm, and the tiles look way more homogeneous. This is the rolling ball algorithm. I think we can look at this over the workshop, but I wanted to keep it here so that you can access it afterwards. Yeah, that's another way of subtracting. And there are other artifacts that are very difficult to correct, or even impossible. So these ones you definitely should do your best to avoid during image acquisition. One of them is bleed through. This happens when you have a couple of fluorophores that the spectra is so close to each other that if you excite, or if you choose the wrong filter sizes, when you excite your sample and you acquire the image, you will see both fluorophores in the same channel. Here we have two rows. One has a simultaneous acquisition and the other one has a sequential acquisition. So in the bottom case, both fluorophores were excited at the same time, and we can see some of the green structures are also appearing on the red channel. If we see the sequential acquisition, only one of the fluorophores was excited during each of the acquisitions of each channel. So we don't really see a lot of the green signal that's on the red channel that we can see down here. And we can already see, if we compose the image, that this one has a lot more tones of yellow because of how the green channel has been bleeding into the red one. This compromises the intensity measurements and the shape measurements. And of course, you won't be able to do colocalization analysis if you have a lot of bleed through. There might be ways of characterizing this bleed through, and then you calculate a sort of a bleed through factor and you can correct the image, but it's not something you can always do. You need to acquire samples always with your same image acquisition settings, and you need to be able to access samples that have either one of your fluorophores present in them. So it's extremely it's very complicated to get. Another problem you might encounter is channel registration, which, I mean, you often need these if you have chromatic aberrations, for example. But then if you see something like this, that you say, okay, these structures are actually supposed to be overlapping with the green ones, well, the first thing you have to do is identify the cause. What was happening here? Was it because you used the wrong media? Is it that you were using the wrong oil? Can you fix it on the image acquisition side? Can you improve the sample preparation? Because if any of these can be addressed, then it's better to do it at this point. That maybe you cannot fix it at this point, and you start wondering, well, can you fix it after characterizing the registration you might need? So you might start to wonder, well, is it a 2D image or a 3D image? There's a lot of questions that you have to answer at this point. You could use beads to characterize what is the registration you need to use to correct this image. But in many cases, if it's because of mismatches, this characterization depends on how deep you go into your sample, and also how much sample you have. between your plane of acquisition and your objective. And this is often very hard to characterize, even impossible. So I really advise you to work on sample preparation before this. And there are some other artifacts that are actually impossible to correct during image analysis. One of them is problems with exposure, for example. If you underexpose or you choose the wrong digital offset, you can see here that the whole background looks blue. So here you have two problems. One, you cannot really estimate your background because it has a single value, so there's no standard deviation here. But the other problem is that if you wanted to segment your image, you kind of segmented it already during image acquisition. So this means whenever you put a different sample there, this background level might be higher or lower, and you're biasing your analysis during your acquisition. And this is information lost that you cannot correct afterwards. Another problem could be saturation. So this is when the values, the intensity values were so high that during acquisition, you cannot really say how high they were. This value could be, it's on top of the maximum. So you don't know how much on top of that it is. So remember to use the high-low or range indicator when you're acquiring so that you see, okay, you shouldn't be seeing the whole background blue. You should be seeing a couple pixels, very, very dim, and try to avoid the saturation, especially on your objects of interest or your regions of interest. And remember that if this saturation is extremely high, it can also cascade into other pixels. Especially, I mean, this happens in camera-based systems, and this renders whole rows of pixels completely useless to estimate intensity or do some analysis afterwards. Okay, let's sum up this part. Improving the acquisition always simplifies the analysis. Some of these artifacts lose information in these images, so we cannot correct for them. Characterizing the imaging system also simplifies how you will correct afterwards. And some corrections might be done via some algorithms, but not all of them. And what we didn't deal with is about spatial filtering, so spatial operations. That's one of the, I mean, there's another interesting part of images, and that there's a lot of information on how the intensities are placed around the image. Here we have two nuclei, and this is a profile of the intensities if you were to draw a line here and make a cut and see what were the intensity values at that line. We can have, for example, a blurring spatial filtering, and then it looks a bit blurred, so we see less details here, but it might be easier to segment, and we removed some of this noise, which was of high frequencies. Or we can do some edge detection. So wherever we see a sharp change in intensity in this image, we will get here a peak. And this is very good to see what are the borders of our objects of interest. How do these work? Well, let's say we have our original image, and then we choose what is called a kernel. The kernels are actually defined by their size in pixels, and then what are the values of these kernels or the operation that is going to be performed. And what we do is we're going to slide this kernel through the image, applying this operation to get what is called a filtered image. Let's see an example to make it easier. Let's say we take a mean filter. So we have this very small image. This is the matrix representing it. And we're going to take this particular pixel and its neighboring pixels, and we're going to calculate the mean, which is similar to multiplying every pixel by 1/9, right? Because we just sum all these pixels and then divide by the number of pixels. We do this and we put that value here. And we're going to do the same for the following pixel, and we put it here. And that's how we're going to do. We're going to use a mean filter. So that was a mean filter. We could also choose, instead of mean, choose a median filter with it, which is good for reducing noise. Let's see a specific example. Here we have an unfiltered image, very small. which has a very bright, what is called like a hot pixel, could be a problem in one of the pixels of the camera. And when we do a mean filtering, what it's doing, it's going to spread out that outlier. It's going to be spread out over 9 pixels. But if we were to do a median filter, that very high intensity outlier is going to disappear. So the good thing about median filters is that we get rid of some of these outliers. So if we have this kind of salt and pepper noise, the mean filter will spread out that noise even more over the image, but the median filter will actually get rid of that. Another typical filter is what's called a Gaussian smoothing, and this is mostly used to blur images. And well, it's called Gaussian smoothing because the shape of the kernel is a Gaussian. This is how it would look if you were to look at it at the intensities in 3D. And one of the good things is that it is computationally very efficient because you can implement it quickly by composing different filters, or you can also use it in Fourier space. And the degree of smoothing is given by this width, by the standard deviation of the Gaussian, let's say. So you can make it more blurred or less blurred depending on what is this parameter that you choose. And what is also very interesting is that you can choose this size in a way to match your point spread function. So this way, you're suppressing noise while also using information that you know should be around the same point spread function. So you're not mixing information from different point spread functions. Well, now that you know this couple filters, you could think about different convolutional filters. You can try to think about some to detect edges. For example, in this case, we have the pre-width operator, which has a negative values here, 0 values in the middle, and then ones to the right, or you can do it horizontal. And you see that these have these are sort of gradients. So these are good to detect and highlight gradients in your images. You can also do your own customized kernel in Fiji, but I really suggest that you look online for different filters, as you might find specific filters for your questions. For example, if you're looking for blobs, blobs or dots, you can use Laplacians of Gaussians for those things, etc. And okay, I'm a bit over time. Let's take a bit of a breather.
We're supposed to go until 10 past, I think.
Yeah. So everything was taking too long for me. So let's jump to the difficult part, which is segmentation, this is the part used to separate our objects of interest from the background. So here we have the sort of different clear steps of segmentation. We want to separate these objects of interest. We start from our filtered image, ideally, or image as an input. And the first step could be to have a semantic segmentation. This is also the result. The output here is called a binary mask. So it has either zeros for what corresponds to background, or ones, or the highest value, for what corresponds to our foreground pixels, the ones that are of interest. And the next step would be to identify different objects in the image. So this is called instance segmentation. And we have a labeled mask where each pixel value refers to the label of a specific object. So all these pixels that are green are probably a 1. All these pixels that are red are probably a 2. This could be a 3. And the background is usually a 0. One of the most basic or known methods is thresholding. We can do this manually by selecting here with a slider, which is our threshold. But we can also choose other automatic thresholding techniques, which are based on the statistics of the pixels. For example, we could choose moments, or Otsu, which is also very well known. And these are automatic thresholding algorithms. They also have their own theory and use cases, so you can look into them to know which one you should be using, or you can try several of them and then decide which one you like and look more into detail of why that one is working and others are not. Otsu, for example, it works by having your histogram of intensities. And what it will look for is what is the value of threshold that maximizes the interclass variance. So if you were to choose an intensity threshold around here, you will have a group of pixels here and a group of pixels here, and the variance, the interclass variance is going to be the highest at that point. Let's say we chose our thresholding algorithm or method, and then we end up with this binary mask. But we say, okay, it doesn't look quite so great. So following, we apply some operation that will fill these holes. This is a piece missing that we cannot really fix at this point. So what we're going to do is we're going to filter by circularity, and we end up with this very nicely segmented nuclei. Think about what you need, what's the information that you wanted to quantify. If you wanted to just count nuclei, maybe you shouldn't have filtered by circularity, because this might not look perfect, but it was still a nuclei that you could have counted. If you wanted to characterize shape, or if you wanted to characterize intensities, then you should have it as clean as possible, but also think that if you want to quantify shape or morphometry, you're filtering by a morphometric property. So you will bias your results. So we can apply these morphological operations to clean up the image. They need what's called a structuring element, which could be a box, a disk, or cube or ball if it's in 3D, and a size in n, a size in pixels. And what we can do is, for example, what's called an erosion operation or dilation operations, and they do what the name actually says. You will take that structuring element and erode whatever structure you found here. So here you can see an erosion of these binary mask here, and this is a dilation, which would be grow it by the size of this structuring element you chose. You can also combine this operations one after the other. One is called closing and the other one is called opening. So one is a dilation followed by an erosion. And what you do in that case, for example, if you have holes and you dilate this, you will fill in the hole. And when you erode it, you will get something very similar to the original shape, but without the holes. And it's cool. It's very good for smoothing contours or fusing narrow gulfs or filling in holes. The opposite operation is also possible. And it's good for eliminating protrusions or bridges or very small things. Once you have your binary mask that you're very happy with, you're going to run, or you could run what is called a connected components labeling, which means every pixel, every positive pixel that is connected, you will assign it the same label. So here we have all pixels belonging to the object 1, all pixels belonging to the object 2, and this is how you end up with a labeled image. Let's say that at some point you had a couple nuclei that were connected or touching each other. So you can run what is called watershed. You generate a map where the intensity value corresponds to how far that pixel is from the border. And then it's similar to if you imagine what would happen if you start filling in basins. If this were basins of empty basins and you start filling them with water, the moment they touch each other, that means you should separate at that point. This is a bit more visual explanation of what is happening here. So I went through a lot of different methods for segmentation and analyzing the image, but nowadays there's been an increase in methods that are based on deep learning. So what is this, the deep learning or machine learning methods? Well, they all belong to the field of artificial intelligence, which are techniques that enable computers to mimic human behavior or human way of thinking. So the classical image analysis that I've shown you up till now usually fall into this artificial intelligence part. Machine learning means that the machine learns the parameters from data that you show to it. So the computer, it's an algorithm that will learn the parameters from the data. While deep learning is within machine learning, but it's usually a specific case where you have neural networks working. So they have a very specific, it is a more specific algorithm. Let's think about a machine learning example where we have an input, which is this image, and what we will do is we will extract features. And when I say extract features, I mean we can choose spatial operations that we've seen before to extract for every pixel what the result after applying that spatial filtering is. And then we will we will label our data, and we will ask the algorithm to learn, okay, which is the best threshold for these things. In these cases, for example, we can use decision trees or support vector machines or random forests that will learn which should be the best threshold for each of these spatial filtered images we generated. If we want to save out a bit more time, we could include the feature extraction within the training of the algorithm. We could use neural networks like convolutional neural networks or recurrent neural networks, encoders. And in this case, we are also training the feature extraction within this algorithm. So you already have a bit of a talk on artificial intelligence on Monday. Let's go to a couple examples here. We can use the what's called the Weka, the trainable Weka segmentation. This is within Fiji. And because we have so many pixels in an image, we can also generate a lot of labeled pixels for training. So let's say we get this drawing tool, and we will paint red the pixels that correspond to these very dark lines here. Everything else should be painted in green, which is background. And we want, I guess this is mitochondria, we will paint them with a line in purple. And the trainable Weka segmentation, or it works similarly to Elastic for the ones that already know about this, it will generate many filtered images with the pixel values for after applying all these spatial filtered images. And then it will train a random forest that will decide, according to all these spatially filtered images, if the pixel should belong to the mitochondria class, if it should belong to the dark membrane class, or if it's just background. Or we could have gone for, we could also combine them to run them in different channels. We can have a trained neural network that segments very well nuclei and a trained neural network that segments very well cells. And we can run this in parallel and then combine this information. We have to always remember to keep test images and to validate and characterize our method to see how accurate it is with our images and with our specific test case. There are a couple of very well-known deep learning models out there, like CellPose, for example, that works very well on several different types of images. It will estimate the flow or how the intensity flows from one pixel to another, and that's what it uses to segment cells afterwards. There's also StarDids that we'll be looking for star convex shape in the images. So if you have many nuclei that are separated in this way, it will try to look for this ellipsoids within the image. And both of these you can actually fine-tune later for your specific case. These are a couple more examples of CellPose. You can use it on blood cells, for example. You can use it on hematoxylin and eosine staining. You can use it on fluorescent images, as I've shown before. There's also SAM. I think the paper came out today of micro SAM, which is this segment anything model. And you should be able to throw almost any type of image to SAM, and it will do instance segmentation on your image. So you're saving a lot of steps here. But okay, I gave a long talk about classical approaches. So why do we still use them? I mean, these I just told you that we save a lot of time by using neural networks. Well, we have what's called a deep learning generalization problem. Not every model works out of the box for every image. So we can see here, for example, we applied one of these models to segment out this image, and either we get the very small nuclei or we get some attempt at the nucleus. There's also a lack of annotated training data. So for each specific case, you might need a data set, a small data set, or labeled data for training. And then we also have to validate and characterize our model or new fine-tuning of the model. And last of all, image analysis is not always limited to just cell and nuclei segmentation. You might want to segment other things. So that's why sometimes using a classical approach is the best way or the fastest way. So let's say we already have our instance segmentation and we want to do our quantification from this. I hope you bear with me for the last couple slides. These are quite fast. So once we have our instance segmentation, we can think about counting the number of objects or measuring some geometric properties or morphometry, studying intensity features, texture features, or relational or features between one another, such as for localization. This is often done in Fiji. If you go to analyze set measurements, you will see a set of different measurements that you can get. Some are shape related, such as the circularity, the roundness, the aspect ratio. This will tell you about how circular or elliptical your object is. You have some other intensity related measurements like the mean intensity or the intensity density, which will sum all the intensities from the image, from the specific objects. You can also do this with the analyze particle tool, and you can get intensity measurements or morphometrics from each separate object. And then in the end, you have this results table that show you what are each row corresponds to a different object in your image, to a different label, and each column will correspond to a different feature. And you can save them as a CSV and then continue your analysis on Excel or on other software of choice. And what about software? Because I just mentioned software. There's a ton of software tools in the bioimage analysis ecosystem. Some of them are custom, some of them are proprietary, some of them are open source, and some of them are very domain specific, and some are very broad or very general. Fiji is quite general. Napari is you would use it with Python, and it's quite general. Elastic is mostly for the segmentation, and QPath works very well for pathology images, and you can go into any of them, but don't hesitate in asking which would be the best for your specific question. And always remember the key point: what is your scientific question? Because if you know what information you have to retrieve from the image, then you also ensure that you have the right quality for it. Remember that, and don't hesitate about turning all these gears around and trying, thinking a bit about image analysis, doing a quick acquisition of a small data set, analyzing it, thinking about if the statistical model or the information is correct. You might notice, okay, you might have to change something and then change the acquisition, change again a bit the analysis, but always start small and start as soon as possible trying out all of these gears. Because if you go to try to attempt a perfect image acquisition and you generate a huge data set, when you jump to image analysis, you might notice, oh, I did not think about this specific thing. And then you generated a whole data set that is almost unusable. I will leave you here with a couple useful links. ImageSC is a forum. It's great for asking questions. It's great for answering things. You will find lots of answers in this forum, and the community is very active. So we will find that we are answering questions there. Here you have a couple books that you might want to check. Analyzing fluorescence microscopy images, or this bioimage data analysis or bioimage workflows. This online book is well, it's online. Everything is clickable. It's quite short, but it's very clear. Some of my slides were based on Robert Haas's Introduction to Bioimage Analysis lectures, so you can click on it and watch his version of the lectures. You can also see some of our material at our YouTube channel. Remember to click like and subscribe as usual. And don't hesitate also in getting in touch with the community. For example, GLOBIAS, which is the Global Bioimage Analyst Society, which started last year. Or you can even check the Bioimage Analyst database that they have here in the GLOBIAS webpage and get in touch with any of us. There you have our emails, and you can ask us for help if you have specific questions or even want to start a project. And I can leave you here with some contact details for us. We have our email where you can ask to start a project. We have a whole plethora of projects you can look into. Blue Sky account for the ones that are more into social media, or LinkedIn of me and Anna Clem, which is the head at the unit. And I guess it's time for a coffee break.
Yes.
Or questions.
Hi, Augustine. Yes, I think what we will do is that we'll shift everything. So don't worry, we'll get your coffee break. We're just going to shift things. So, do you guys have questions now to Augustine?
Yes, I have one. When it comes to the post-processing, like the masks or the where you would fill in the holes with the erosion and the dilation, I'm thinking, do you know when you go to a natural history museum and you see in the dinosaurs, they have the bones or the fossils that they filled in, colored differently to the ones that they found. Is in the same sense, is it worth showing these steps where you filled in parts that weren't there beforehand? Because I rarely see anything like that, and I think it would change because it's up to interpretation, I guess, when you fill in stuff versus when you don't. Um, how much of these steps should you show?
So yes, morphological. It's almost an art or a handcraft, because you can combine them one after the other and a lot of times. So it changes your interpretation. You have to be very careful with them. Sometimes you just spend hours combining an erosion to get rid of the small things, then a dilation to try to fix what you just eroded, and then you think, oh no, but I would like to connect some stuff that was not segmented properly because I was missing some things. Yes, it is a handcraft. I don't have a specific. Oh, this might be an interesting example for that. You could go very far with dilating this object so that you finally fill in this hole here and then erode it again. But you have to be really careful, because if you do that too many times, everything will end up being the structuring element you used. So if you were using a circle here and you dilate everything a lot and then you erode it a lot, you end up with things that are just circles or perfect ellipses.
When doing things like this, what happens is that you work for hours on this image, and then the next image doesn't work at all with the same. So it's important to test the analysis as you're going on many types of images. And that's why you need to have a pipeline that you can easily apply so that you can judge.
Yeah, and it's the other key factor is don't aim for perfection. You will always have to sacrifice something.
Makes sense. But then about showing them, like, do you show these as well? Like, is it possible to show the segmentation steps that you took?
Yes, yes, you can show them. I don't have one here, but if you the easy way of showing them is if you apply, let's say you have the original image and you apply an erosion. you can subtract one image from the other, and then you get the outlines, and that's the actual change you did on your image. So you can easily show what was changed from one image to another. So by applying subtraction, you can show what were the differences.
Thank you.
Or even summing, and then you have 3 levels or 4 levels. You can be very creative with all these things.
Yeah. Okay. Thank you for the advice.
Okay.
More questions? Ken?
Yes, I have a question about segmentation because I use a cell pose to segment my samples. But maybe I can copy the image in our chat. Maybe you can have a look. It's oh no, I cannot. But
Should I stop sharing?
Do you want to share, Chen?
Yeah, I want to share my screen. Maybe.
Right.
Yeah. Remember, it's being recorded, right?
Hey, it's beautiful.
Yes, I record it. Yeah. Yeah, can you see that?
So we can see, yeah.
Yes.
Yes, it's like I use a cell pose to do segmentation on the organoid slides, but the result is a bit weird. For example, there are no intact cells on the edge of the organoids, but sometimes it put mask on that. But sometimes, look at here, there is a cell here, but there's no mask on that. So it's like the result and the real situation, they are different. So how can I adjust that?
So you can always
Can I just say something? Sorry.
Yeah.
I'm just wondering if we could take this during the workshop because it's quite a specific case and maybe people want to have a little break. We're a little bit late. Is it okay, Chen, if we take this
Yeah,
during
it's
the
okay.
workshop instead? Yeah?
Yeah, it's okay.
If there are no extra burning questions, Augustine is also going to run the workshop, so he's staying with us. Let's go for a break for 10 minutes. So we come back at 32 in the workshop Zoom. Okay?
Yes.
You guys.